Hello all, welcome to my presentation on the topic Schistosoma or blood flukes, and in this video I will discuss about Schistosoma hematobium, Schistosoma mansoni and Schistosoma japonicum, and I ensure you guys that at the end of this lecture you all will be able to recall everything I am taking in this class, so please be attentive. So Schistosoma are blood flukes, they are parasitic flatworms. They are flatworms or platyhelminthes, and they are actually trematodes. Flatworms can be classified into cystodes and trematodes, and Schistosoma belongs to trematodes, and they are blood flukes which are found in the blood. Actually, the Schistosoma, they lives in venous plexuses. So that's why they are known as blood flukes. They live permanently inside the veins of their definitive host, which are humans. Now, why schistosomes are unique trematodes? Unlike all other medically important trematodes, schistosomes are dioecious. They have separate male and female sexes, and all other flukes or trematodes are hermaphroditic. The schistosomes are dioecious. Okay, this is a unique feature of schistosome, and they live exclusively in venous plexuses, and their eggs are elongated and non-operculated. The eggs of the schistosomes are elongated. Okay, so operculated means other trematodes they have a lid called a operculum on their eggs, and the schistosomes eggs are non-operculated and they are elongated. And the schistosomiasis is the disease transmitted by schistosomiasis, and the infection occurs via skin penetration. Okay, skin penetration by the larvae cercariae. Okay, actually schistosomes has adult worm which lives in the humans, and they have eggs which are passed through the feces or urine of humans, and they have larvae. Larvae are mainly miracidium larvae. Then from miracidium sporocyst develops, and from sporocyst the cercarial larvae develops. The schistosoma has these three larvae, and this cercarial larvae is the infective form for humans, and they penetrate the skin. By skin penetration they causes schistosomiasis, and schistosomiasis are different types. I will come to that. And they have only one intermediate host, which are freshwater snails. Okay, so these are the features that makes the schistosomes a unique trematode. And the schistosomiasis is the disease caused by schistosomes, and these can be urinary schistosomiasis and intestinal schistosomiasis. Okay, the urinary schistosomiasis is caused by Schistosoma hematobia. Okay, and the intestinal schistosomiasis is caused by Schistosoma mansoni and Schistosoma japonicum. Okay, you have to remember this. And now let's discuss the morphology of each. So we are going to discuss about these three schistosomes: Schistosoma hematobium causing urinary schistosomiasis, and Schistosoma mansoni and japonicum causes intestinal schistosomiasis. Come to morphology, and morphology is general for all the three schistosomes, that is hematobium, mansoni and japonicum. They have I already told about morphology, that they have adult worm and they have eggs. From adult worm, the adult worm lays eggs, and inside the adult worm, inside these eggs they have larvae, and they have three larvae. From the egg hatches and releases larvae. Larvae are miracidium, from which sporocyst larvae develops, and From the sporocyst, the cercarial larvae develops, which is the infective form in humans. So the adult worm, coming to the adult worm, the morphology is general for these three species. The adult worm is leaf-like and they are unsegmented with two cup-like suckers. We can see two suckers for the adult worm. They have anterior sucker and ventral suckers, and they have a digestive tract. They have complete digestive tract, and the intestine is bifurcated. The intestine is inverted Y shape. You can see bifurcation of the intestine. Okay, and I already told you that they are dioecious. They have separate male and females, which is a unique feature of Schistosome 7 trematodes. And the males are broader. You can see the males are broader. This is the male. This brown colored one is males, and males are broader, and the females are slender and long. This is the female. This blue one is the female. Females are slender. So females are slender and males are broader, and the males have a gynecophoric canal in which this female worm lives permanently. Okay, the female lives permanently inside male's gynecophoric canal. The males are broader and thicker with the gynecophoric canal. The females are long, slender and cylindrical, and the female lives permanently inside the male's gynecophoric canal. So the adult worm morphology is similar for the three types: hematobium, Mansoni and Japonicum. Okay, and the egg morphology is different for these three. It is the egg morphology that helps to differentiate between these three species. The hematobium I already told you that eggs are elongated and they are non-operculated. They does not have any lid-like structure. Okay, eggs are elongated and they have spines. And this position of these spines is which helps to differentiate between these three species. The hematobium has a terminal spine. You can see a terminal spine. Hematobium has a terminal spine, and the Mansoni has a lateral spine. You can see hematobium has terminal spine, and the Japonicum does not has any spine. They has a small lateral knob. Okay, Japonica has small lateral knob. Okay, you must remember this. This is very important. So you can remember it as like HTML. That is, hematobium has terminal spine, Mansoni has lateral spine, and Japonicum. Japan is a very small country, so they have small lateral knob. Okay. Now the third form is larvae, and larvae are three forms: Miracidium, then sporocyst, and then the cercarial larvae. Now coming to the life cycle of schistosomes. Life cycle, the definitive host is man, the intermediate host is freshwater snails, and the diagnostic form is non-operculated eggs which are passed in the urine or feces of humans. The eggs are passed in the urine or feces and they are non-operculated. Okay, so we diagnose this condition mainly by microscopy of the urine or stool, which shows characteristic eggs. Okay, the infective stage for snails is Miracidium. The eggs are passed in the stools or urines. Okay, urine or stool, and these eggs releases the larvae, Miracidium larvae, and this larvae is infected to the snails. Okay, it infects the snail, and in snail the Miracidium develops into sporocyst, and the snail releases it as cercarial larvae into water. The snails are in freshwater, and the snail releases cercarial larvae in water, which infects human by skin penetration. Okay, so the infective stage for humans is cercarial larvae, which has a forked tail. Okay, for snails the infective stage is Miracidium, and in humans the Schistosoma. I told you that in humans the Schistosoma lives in the venous plexuses, and Schistosoma hematobium specifically lives in the vesical venous plexus. vesical venous plexus. Okay, venous plexus of urinary bladder, that is vesical venous plexus, and they causes the condition schistosoma, urinary schistosomiasis. And the Manzoni and Japonicum lives in the mesenteric venous plexus, and they causes intestinal schistosomiasis. Okay, now let's discuss the life cycle in detail. The humans lays eggs which are shed from which are passed through the feces, and in feces the eggs of Manzoni and Japonicum are seen, and in urine the eggs of Schistosoma hematobium can be seen, and this eggs hatches and releases the miracidium which penetrates the snail, freshwater snail, and in the freshwater snail the sporocysts develop in snail, and they develop again and the sporocysts are released into the freshwater as cercarial larvae. They are fork-tailed. Okay, and the cercarial form penetrate this human skin to become schistosomale. The cercarial loses its tail and becomes schistosomale, and which through the circulation reaches the portal blood in liver and matures into adult. Okay, in the liver they matures into adult, and these adult migrates to specific venous plexuses, that is mesenteric venous plexuses for Schistosoma, Manzoni and Japonicum. In the mesenteric venous plexus, and the Schistosoma hematobium migrates to vesical venous plexuses. Okay, so the pathogenesis and clinical features. Actually, all three forms causes cercarial dermatitis that occurs within hours or days after the skin penetration by the larvae, cercarial larvae, and there is itching at the penetration site. Okay, and the papular rashes and local redness can be seen, and this condition is known as swimmer's itch. Okay, cercarial dermatitis. And next is acute schistosomiasis. Okay, so first clinical feature is cercarial dermatitis. The second is acute schistosomiasis, and third is chronic schistosomiasis. Okay, cercarial dermatitis is similar for all three species. The itching at the penetration site, papular rashes, and local redness. Okay, and the acute schistosomiasis is called Katayama fever, which coincides with the egg laying. Okay, actually it is a type 3 hypersensitivity reaction to the egg antigen, and it presents as fever, chills, then hepatosplenomegaly, then headache, then myalgia. Okay, some vague symptoms are seen, and also dry cough. All these symptoms are seen. Eosinophilia. These are included in acute schistosomiasis. They occurs 2 to 8 weeks after the infection. Okay, and chronic schistosomiasis can be again classified into chronic intestinal schistosomiasis and chronic urinary schistosomiasis. And chronic intestinal schistosomiasis is caused by Schistosoma Manzoni and Japonicum, and urinary is caused by Schistosoma hematobium. Okay, chronic intestinal schistosomiasis. It has both intestinal and hepatic manifestation. Intestinal manifestations are abdominal pain. Okay, intestinal symptoms like abdominal pain, and then chronic diarrhea, blood and mucus in stools, colonic ulcerations, and hepatic manifestations include periportal fibrosis around the portal system. There is fibrosis, and then portal hypertension can occur due to obstruction of these portal veins by adult worms. Portal hypertension can occur, periportal fibrosis can occur, and due to portal hypertension, ascites can occur, then splenomegaly can occur, esophageal varices can occur due to portal hypertension. Okay, and hepatosplenomegaly can be seen. Next is chronic urinary schistosomiasis, which is caused by Schistosoma hematobium. While intestinal is caused by Manzoni and Japonicum. Okay, so urinary schistosomiasis, you can see a terminal painless hematuria. Hematuria means passage of blood in urine, and it is often terminal. At the end of urination, a painless hematuria can be seen, which is characteristic feature of urinary schistosomiasis, and increased urinary frequency, suprapubic pain, obstructive uropathy due to obstruction of urinary tract by adultum, obstructive uropathy, which causes hydronephrosis. Hydroureter means excess water in accumulation in ureter and excess water accumulation in kidney is called hydronephrosis, and that can further lead to renal failure. And also schistosoma is one of the parasites that can cause microbial carcinogenesis, and it can cause squamous cell carcinoma of bladder due to chronic inflammation. Okay, this is about chronic urinary schistosomiasis caused by Schistosoma hematobium. Now diagnosing schistosomiasis. Okay, diagnosis the gold standard is microscopy. Okay, microscopy is the gold standard for diagnosing schistosomiasis, and microscopy we usually do microscopic demonstration of egg, and these eggs are passed in feces in case of schistosomiasis due to Manzoni and Japonicum, and eggs are passed in urine in schistosomiasis by Schistosoma hematobium. So if we suspect a patient with schistosomiasis, we can do microscopy of feces or either urine, and that will show characteristic eggs which are elongated and non-operculated, and they have spines, terminal spines for hematobium, lateral spines for Manzoni, and lateral knob for Japonicum. Okay, and also we can do serology. In serology, we can do ELISA. We can do antibody detection by ELISA or immunofluorescence test. Okay, we can do detect antibody, and also we can do antigen detection. Can detect the antigen of the worms, which are CCA or CAA antigens. These are two types of antigens of schistosoma. We can detect these antigens for diagnosing. Okay, and also next is histopathology biopsy. We can do rectal biopsy, then we can do bladder biopsy. Okay, and in biopsy we can see egg surrounded by granulomatous inflammation. Okay, this is you can see egg surrounded by granulomatous inflammation. Okay, and also we can do x-rays and ultrasound for detection. Okay, so if we suspect a patient for schistosomiasis, we can first do urine or stool microscopy for detection of specific eggs. And if the urine or stool is negative, but we still suspect the schistosomiasis, we can do serology or antigen test. Okay, serology antigen test to detect antibodies or antigen, and we can confirm the findings by PCR or biopsy. I missed PCR. Okay, PCR is a molecular method for detecting parasite DNA in urine or stools. Okay, we can detect parasite DNA in urine or stools by PCR. So we can confirm by PCR or biopsy, tissue biopsy. First gold standard is always stool or urine microscopy. Now treatment and prevention. The drug of choice is praziquantel, and alternate drugs include these metryphenate, oxaliplatin, and for prevention we can do snail eradication. We can prevent environmental contamination, avoid infected fresh water. And this is the final differentiating features, a tabular column. You must remember this. Okay, so habitat for Schistosoma hematobium is vesical venous plexus, and it causes urinary schistosomiasis. For Manzoni, habitat is inferior mesenteric venous plexus, or Japonicum it is superior mesenteric venous plexus. Okay, for Manzoni and Japonicum it is mesenteric venous plexus, and for hematobium it is vesical venous plexus, and Mansoni more specifically inferior mesenteric venous plexus, and for Japonicum superior mesenteric. Okay, and egg shape, the for hematobium has terminal spine, HT. Hematobium has terminal spine. Mansoni has lateral spine, HTML. Mansoni has lateral spine, and Japonicum has small lateral knob. Japan is a small country, so Japonicum has small lateral knob. This is only a mnemonic. Okay, and the hematobium and Mansoni are not zoonotic. These are mostly affecting humans, but Japonicum mostly affects the animals. Okay, so it is a zoonotic infection, and hematobium causes urinary schistosomiasis, and Mansoni and Japonicum causes intestinal. So Mansoni and Japonicum are almost similar, and hematobium causes squamous cell carcinoma of bladder in long run, can lead to renal failure and all, etc. So that's all. Thank you.